Hi, my name is Vanita Giddings, and I am an educator at the Math Science Innovation Center. I'd love to welcome you to my virtual lab. Today, we are going to be doing spinach chromatography. Now, chromatography is a process by which we separate mixtures. The mixture that we are going to make today consists of spinach leaves and 100% acetone. Now, you could also use isopropyl alcohol. And so let's go ahead and get started with our experiment. I'll tell you what you'll need for materials. So, of course, you'll need spinach. Okay, so you'll need to have spinach leaves. Now, if you don't have spinach leaves, that's okay. You can do this chromatography experiment with maybe kale that you have in the refrigerator, or even leaves that you gathered while you went on a hike or a walk in your neighborhood. You are going to need coffee filters. You are going to need a glass, maybe two glasses. You will need scissors. You will need a spoon, or you could even use a popsicle stick. You'll need pencil. You will need some measuring spoons. The 100% acetone, again, you can use isopropyl alcohol if you'd like. I believe that's it. So to start, we're going to make our extract. And so what we'll do is we'll use our scissors to cut up these spinach leaves. And you cut them up as finely as you possibly can. And then you put them into the cup. So here you see I've already cut up some spinach leaves. Okay, really finely. And then for this particular glass that I have, and it should be a glass, not a plastic cup, because the acetone can eat through the plastic cup. I use about 2 tablespoons of the 100% acetone. You don't want this acetone to splash at all. And so that's why I was telling you to use measuring spoons. Now I have a 2 tablespoon measuring spoon, and I found that that is enough to put into my glass in order to cover the spinach leaves that I've cut up. I don't cut up more than about 2 or 3 spinach leaves. You want to pour the acetone kind of off to the side so it's not going to splash, all right? And you don't want to have it near your face. Now, you will then let that sit for about a half an hour. Now, when I did this earlier, I took mine and put it outside on the patio, okay? Because, of course, it's acetone, or alcohol if you choose to use alcohol, so it's going to have a fume. If you don't want to sit it outside, maybe there are others that might get into it or a pet, then you could actually go ahead and cover it with something. The next thing that we're going to do is to take one of the coffee filters, and we're going to open that coffee filter up, okay? And then after opening it up, you're going to fold it in half. We don't want to put our fingers all over the coffee filter a whole lot because of the fact that there are oils on our fingers, and those oils will impede or stop the movement of the pigments that we're going to be looking at from moving from one point on the paper to the next, okay? So in putting the acetone and the leaves into the glass and letting them sit for 30 minutes, we are basically extracting the pigments, the color from the leaves, all right? And so yes, leaves generally are green. However, there are lots of other colors inside those leaves as well, or pigments as they are called, inside the leaves. And we want to be able to see those various pigments, and also we want to know how far those pigments can travel. And so we're going to get into more of an understanding of that in a little bit. So let's go back to our paper, our coffee filter. So what I'm going to do is I'm going to cut long strips straight across, okay? And then after doing that, it's pretty thick, so I don't need a filter piece of paper that long, that thick, so I can cut it in half once more. Okay, we'll cut it in half. Doesn't have to be perfect. And so this is still pretty long, all right? So if you had a tall glass, you could use this, but for the glass that I chose, as you can see, it's a smaller glass, all right? Then I chose to cut my piece of paper in half. So this is basically the chromatogram. If you were in the lab, you would actually use chromatography paper, and it'd be really important that you didn't put all your oils all over it, because again, that would impede the movement of the pigments. All right. Now, once we have this chromatogram, our little coffee filter, okay, then we're going to take our ruler and we're going to mark from the bottom about 2 centimeters. Okay. We're going to use our pencil and mark 2 centimeters, and we're going to draw the line horizontally. In the middle of that line, you'll be able to draw like a little circle. That little circle is where you're going to put your extract right there. All right, you can see the line here. It's 2 centimeters from the bottom of the paper. There's a horizontal line that I made, and you're going to put the pigment right across that line. In the other glass, we'll put 2 tablespoons of the acetone or alcohol, and we'll let that sit just before the 30 minutes is actually up. You can make your strip, your chromatogram, long enough so that it can be, you can bend the paper over the rim of the glass, okay? You could tape the chromatogram to the side of the glass, or if you wanted to make it longer, you could even tape the chromatogram to a pencil and then put it on top of your glass, okay? The whole purpose of this is that you only want to get about 1 centimeter of the paper wet, and it's going to act as a wick. And so that solvent, the acetone, is going to move up the paper. So I use a scientific word, right? Solvent. Solvent is a part of a solution that does the dissolving, okay? So in this case, it's the acetone or the alcohol, the isopropyl alcohol, okay? The solute is the portion that is to be dissolved, the part of the solution that is to be dissolved, okay? And so with that, we actually have the spinach extract that we would have waited 30 minutes to get. After the 30 minutes, after it's set, then you can take maybe the handle of the spoon and take some of the liquid and put on that dot, or you could put it all the way across that line horizontally, okay? You can do that, or you can get really creative. You don't have pipettes at home, and maybe you don't have an eyedropper at home, but you may have a little coffee straw that you could use. And if you touch the top of it, the straw, and put the straw down into the liquid, then it will move the liquid up into the straw, and then you'll be able to deposit the extract onto the paper, okay? I even have tried using the little, I use eye drops, and I rinsed out one of the little droppers pods that I have, and I was able to suck up or suction up some of the extract, and then I was able to put it onto the paper, all right? Now, once you put your extract onto the paper again, you're going to put it into your glass, okay? So because of the length of the paper, the way that I cut it, I'm just going to fold the paper over the side, and I'll let it sit there for about 20 minutes. And we should see different banding patterns of the pigments or the extracts. All right, so let's go back a little bit. Let's learn about what pigments are, or the purpose of these pigments. We know that they're colors, but we want to know the purpose of the pigments. Again, the pigments are basically the colors that you find in the leaves, okay? There are lots of them. The main pigment is chlorophyll A. Chlorophyll A, there's also chlorophyll B, I guess it's second in charge. Chlorophyll A is bluish green, and chlorophyll B is a yellowish green color. And then there are the carotenoids. Those are the xanthophylls, carotene, and even phaeophytin. So these are all pigments that are found in various leaves, and there are many, many more. So again, chlorophyll A is the main pigment. Notice that on this slide, it tells you that these are photosynthetic pigments. So what the pigments actually do is they absorb light. Now there's a lot of different wavelengths of light, and so chlorophyll A absorbs at a specific wavelength or absorbs a specific type of light. Chlorophyll B absorbs at a specific wavelength or absorbs a specific type of light. What is photosynthesis? I bet you said that it's the plant's ability to make its own food, and that is absolutely correct. Move on to the next slide. Now, the light that is absorbed by these pigments is actually white light. White light contains all of the different colors of the rainbow, and so those different wavelengths or different colors of light, again, are absorbed by different pigments. And so this is a very, very efficient process because we have this white light and we have all of these different pigments that are absorbing all of the different parts of the white light. And so they're gathering as much energy as possible. Carbon dioxide, water, and the presence of solar energy will allow for the plant to make its own food. That food consists of glucose. In addition to that, the plant will give off oxygen, which is pretty awesome for us. And the pigments kind of move up, vertically up the coffee filter. We can see different bands of pigments. So if you're using baby spinach, like I did, you might get all of these banded patterns. But if the baby spinach isn't fresh, then you might not get the carotenes. If you use some other leaf, you might get some other accessory pigments other than the xanthophylls and the carotenes. At the end of this, you'll have a chromatogram, and again, that's just a record of the pigments that were found in that particular leaf that you experimented upon. And you're able to take that on a sheet of paper and you can write on it and show where the different bands stopped. You can also mark on there where the solvent ended. That's very important as well. Thank you so much for taking the time out today to hang out with me in my virtual lab. I hope that you've learned a lot about the chromatography process, about pigments, about photosynthesis, and I hope that I was able to inspire you to go and kind of learn a little bit more, all right? I have made a presentation, create a presentation for you, as well as shared some procedures. You wanted to look at a couple of different procedures and an extension activity. So if you want to add to this or change it in some way, you are more than welcome to do so. All right. So there's so many things that you can do and so many things that you can come up with in STEM, in science. It's all about asking a question. I hope that you come up with some awesome questions from today's experience. Have a great day.